the plasma membrane. Every cell has a cell membrane made up of phospholipids, which are made up of a negative head, which is hydrophilic, and a nonpolar uncharged tail that is hydrophobic. A phospholipid bilayer is formed with tails on the inside and heads on the outside, so the tails are protected from the extra and intracellular fluid. They also provide a boundary between the interior of the cell and the outside environment, as well as control the transport of materials in and out of the cell. Items that can pass through the lipid bilayer are small nonpolar molecules, such as oxygen, CO2, small polar molecules, such as water, large nonpolar molecules, and the main thing about water is that they need aquaporins, and aquaporins are proteins that speed up the transport of water. Otherwise, water would probably take much longer than needed to be able to support life. Then we have items that cannot pass through the lipid bilayer, which are large polar molecules. This includes monosaccharides and amino acids, and ions. What this means is that they would actually have to go through transport proteins, which assist in the passage by allowing the molecules to avoid the hydrophobic portion of the cell's membrane. Next, we have the fluid mosaic model, which is a constantly moving sea of phospholipids that have embedded proteins, carbohydrates, and cholesterols all throughout it. They're held together by hydrophobic interactions, which are actually weaker than covalent bonds, but allow lipids and proteins to shift along the bilayer surface. Cholesterol is unbelievably important in this, and are only found in the bilayer of eukaryotic cells, which helps to regulate fluidity when under different environmental conditions. So when it tends to be in a too warm environment, it will actually bring them together, but then when it's in a too cold environment, it'll try and separate them. And this is because in a colder environment, the phospholipids will try and stick together and not move, which is exactly the opposite of what they should be doing. So in order to make sure that they continue to move, they have to break them up. Finally, we have carbohydrates, which can come in the form of glycoproteins, which are attached to embedded membrane proteins, or glycolipids, which are lipids with one or more carbohydrates attached to them. Here's a more detailed look at the phospholipid bilayer, as well as a diagram of what a phospholipid looks like. Finally, we have the embedded proteins. For the integral proteins, which is one type of the embedded protein, they span the entire membrane and are hydrophilic with charged and polar side groups, but hydrophobic with nonpolar side groups that penetrate the hydrophobic interior of the phospholipid bilayer, also known as transmembrane proteins. This makes them amphipathic as well because they fit the criteria of both hydrophobic and hydrophilic. Finally, we have the peripheral proteins, which are loosely bound to the surface of the membrane and are considered hydrophilic with both charged and polar side groups. The final thing to consider is that proteins that are near each other are often functioning similarly. So this is why integral and peripheral proteins are found so close together. Thank you so much, and I hope this helped.